Hi everyone, today I will be discussing the process of mitosis and meiosis. So, mitosis is when the cell splits apart. Mitosis eventually cells needs to duplicate. There are two main methods of replication. It is the mitosis and the meiosis. So, mitosis is the big idea to remember is that mitosis is the simple duplication of a cell and all its part is duplicate. DNA and the two new cells' daughter have the same pieces and genetic code to identical copies came from one original. Beyond the idea that two identical cells are created, there are certain steps to process before our cells divided in its resting phase. The cell in resting phase is not actually resting, since it has to maintain its reservoir before it undergoes division. This is called interphase. Interphase, it prefers the cell for division. Protein synthesis, DNA duplication, and enzymes after this phase comes from mitosis, which is further divided into two: karyokinesis, division of nucleus, and cytokinesis, division of cytoplasm. Karyokinesis involves the four basic steps of mitosis that are prophase, metaphase, anaphase, and telophase. Here we start the prophase. Prophase is the chromosomes start coiling and become condensed nuclear envelope. This integrates gene transcription ceases during prophase and does not resume until late anaphase, the early G1 phase. The nucleus also disappears during early prophase. Centrosome gives rise to mitotic apparatus, generation of spindle tubules, and metaphase. Now all the pieces are aligning themselves for the big split. The DNA lines along the central axis. So, anaphase, here we go. The separation begins. Half of the chromosomes are pulled to one side for the cells. Half go the other way. When the chromosomes get to the side of the cell, it's time to move on to the telophase. Telophase, now the division is finishing up. This is time when the nuclear envelope begins to desynthesize again along the separated chromosomes. Cytokinesis pinches the cell membrane in the middle of the membrane, closes in, splits in the cell into two. The next important phase of cell cycle is the meiosis. Meiosis, have you ever wondered why you are different from your siblings? The answer lies within the amazing process of meiosis. So what is meiosis? Meiosis is a process where a single divides twice, produce four cells continuing of original amount genetic information. These cells are our cells. Sperm in males and egg in females. During meiosis, one cell divides twice from four daughter cells. These four daughter cells only have half the number of chromosomes than the parent. They are haploid. Meiosis take place a germ in sex cells called gametes. Eggs in females and sperm in males. Similar to mitosis, cells also pass through the interphase G1S and G2 phase. Here are the distinct phases of meiosis. The interphase, meiosis 1, cytokinesis 1, meiosis 2, cytokinesis 2, first at the interphase. So, interphase, DNA is copied resulting into identical full sets of chromosomes. To prepare for division outside of the nucleus are two centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. The next step is the Meiosis One. Meiosis 1 takes place for following steps: prophase 1. Prophase 1 is typically the longest phase of meiosis. During prophase 1, homologous chromosome pairs and exchange DNA created during crossover. Crossover are the significant source of genetic variation and result for new combination of alleles, which may be beneficial to the paired and replicated chromosomes are called valence of tetrads. Which have 2 chromosomes and 4 chromatids, with 1 chromosomes coming from each parent. The process of pairing the homologous chromosomes is called synapsis. At this stage, non-sister chromatids may cross over the point called chiasma. In plural, chiasma in singular. Leptin is the very short direction when the progressive condensation zygote chromosomes approximately line up with each other in the 2 homologous chromosomes of the normal complex. The paired chromosomes are called vibrant or tetrad. These chromosomes pocketing. This is a stage when homologous recombination, including the chromosomal crossover, crossing over. Non-sister chromatids in metaphase 1. Homologous pair move together along the metaphase plate. Paired homologous chromosomes align along the equatorial plane that base the spindle. Anaphase 1 kinetochore microtubules shorten pulling homologous chromosomes, which consists of a pair of sister chromatids. Telophase 1 is the first meiotic division effectively ends with the chromosomes arrive at the post, no DNA replication during the stage. In interphase 2, meiosis 2 is the second meiotic division and usually involved in equation segregation or separation of the sister chromatids. The 4 main steps of meiosis are called the prophase 1, metaphase, prophase 2, metaphase 2, anaphase 2, and telophase 2. In prophase 2, we see the disappearance of the nucleus and the nuclear envelope again, as well as the shortening and thickening of the chromatids. In the metaphase 2, centrosomes contain 2 kinetical that attach to spindle fiber from the centrosome. At the opposite poles, this followed by anaphase 2. In anaphase 2, in which the remaining centrometric cohesion is cleared, allowing the sister chromatids to segregate. The process ends with telophase 2, which is similar to telophase 1, and it's marked by disassemble the spindle and the condensation of lengthening and of chromosomes. Nuclear envelope reforms and cleavage or cell plate formation eventually produces a total of 4 daughter cells, each with a haploid of chromosomes.